Hey, it's Miss King. I'm going to go ahead and do some Punnett Squares with you because I can't really find a good YouTube video that I like. So I'm just going to do some simple Punnett Squares with you guys today on my dry erase board to try to get you to kind of learn how to do them. So we're going to start with something pretty simple, and that is going to be the gender of offspring. And the gender of the offspring is represented in humans on the 23rd chromosome, and we represent gender using X's and Y's. So for a female, you're going to be XX for your 23rd chromosome pairing, and for male, you're going to be an XY. So the Y is what makes you a male. The Y is what's telling your body to not form into a female, but to form into a male instead. So what we're going to do is we're going to take these and we're going to figure out what is the percentage of chance of you having a male or a female offspring. So we're going to use a very simple Punnett square that looks like this. Okay, so this is your basic Punnett square. And how we use the Punnett square is we're going to take our alleles, so in this case, XX for female, XY for male, and we're going to plug them into this, this grid here, and then we're going to figure out what is our percentage of chance. So we're going to start with the female, and we're going to actually put the female's alleles up here on top. So you can see I have the two X's. I've split them up. One X goes on top of one square, and one X goes on top of the other square. And next, I'm going to do the males, X, Y, and you can see that I've split up the two alleles here and here. Okay, so this is your basic Punnett square. Now, all you have to do with a Punnett square is you just have to take your alleles and you just got to drag them down or drag them across. Okay, so we're going to start with this one first. This X is going to go down. It's going to go here and here. Boom, boom. Okay? This X is going to do the same thing. It's going to go here and here because it's being pulled down. So they're just being pulled down into the grid spots below them. Next, we are going to do this X here. It's going to get pulled across here and here. And then next, the Y is going to get pulled across this time, and it's going to be pulled across this way. Okay? Now I did them in two different colors just to kind of help you see where they came from. So what we get left with is we have two of our four boxes have the two X's in them, which is female. So two boxes are female. And two of our boxes, this box here and this box here, are two male. Okay, so it's 2 and 2. If we have four boxes and two of the boxes are females, that means that we have a 50% chance of getting a female offspring. And if two of the boxes of the four boxes are male, we also have a 50% chance of getting a male offspring. So that's why it's 50-50 for male and female. When the child is first, the fertilized egg first happens, you either get an X and an X or an X and a Y. 50-50 chance all the time. So here's our first one. It's the very basic Punnett square, and we use the male and female phenotypes. So female would be the phenotype for this one. XX would be our genotype. Male would be the phenotype, or the physical traits of this one, and its genotype would be XY. Our offspring in our Punnett square, we have a 50% chance of having a female, which would have the XX chromosomes. We have a 50% chance of having a male, which would have the XY chromosomes. So there's your first basic Punnett Square. Hope that was easy to follow. I'll do a couple more for you in a couple minutes. All right, for my next example, I'm going to use this as our question, and I'm going to show you how to put it together to get the answer. So what we're going to do is we're going to cross a heterozygous tall pea plant with a homozygous short pea plant. Now I'm using some vocabulary words from our the slideshow that I did last week, so let's go over those to start. So we're crossing two different types of pea plants. First one that we're going to talk about is this term right here, which it says heterozygous. And if you guys hopefully remember, heterozygous means it has two different letters with it. So it'll have a capital and a lowercase letter. So heterozygous, capital and lowercase letter, we're using tall as our phenotype so that we know that it's a tall pea plant. And to get our genotype, because it says heterozygous, we already know that it's going to be the mix between the two upper and lowercase letters. So this tall pea plant is going to be represented by a capital T and a lowercase t. Okay? So our heterozygous tall pea plant is going to be represented by a capital T and a lowercase t. The second part says that we're going to cross that pea plant with a homozygous short pea plant. Okay? Now homozygous means you have the same of the two letters are going to be the same, so you're going to be either going to be both capital or both lowercase. In this case, the tall pea plant is the dominant trait, being tall. So when it says homozygous short, that means we know that it's going to be represented by our two lowercase t's because it is the recessive trait. Okay? So I've taken our word problem here and I've now told you that we have our tall pea plant is represented by capital T, lowercase t, and our short pea plant is represented by lowercase t, lowercase t. So now we are going to cross these two using our handy-dandy Punnett Square again. So I'm going to go ahead and put the Punnett Square on here. Try to make sure not everything falls over. And I'll revisit this, which is these are the parents that we're going to be using. Those are the genotypes for the parents that we are using. I'm using color on this just to help represent for everybody so it's a little bit easier to follow along with. So we're going to take our two parents. We have a capital T and a lowercase t for our tall plant. And we have a lowercase t for our short pea plant. So we're going to take our alleles right here. We're going to split them up. So we're going to take the capital T, we're going to put it above that spot, and the lowercase t is going to go there. Then we are going to take our short alleles, our lowercase t's, and we're going to put them on this side over here. Okay? Now, again, just like I did before, we're going to drag down and we're going to drag across. So we're going to start with our capital letter, which is this here, and we have a capital T. That capital T is going to get dropped down to both of these boxes. Okay, again, remember they get dropped down. Then our lowercase t is also going to get dropped down into these boxes here. Then we're going to take our short pea plant alleles and we're going to drop or pull them across. So you can see this one's getting pulled across. And this one will get pulled across as well. So we are looking again. We have 4 squares. We have 3 squares. We have 2 squares, sorry, that have the capital T in them. Okay? We have 2 squares with the capital T, which means 50% of these offspring will be tall. And 50% will be short. All right? So this one looked very similar, or turned out very similar to the other one that we did. But in this case, we have too tall and too short. All right, so I changed it up just a little bit for the next one. Now we're doing the same thing with the top here. We're crossing a heterozygous tall pea plant with another heterozygous tall pea plant. So both of these are heterozygous tall. Both of these are heterozygous tall. So they're going to have the same lettering. They're going to be capital T, lowercase t, and we're crossing it with a capital T, lowercase t. Okay? So let's go ahead and do that one. Again, we set up our Punnett Square. We know that our two parents are going to be capital T, lowercase t, capital T, lowercase t, which means they're both tall. But we're still going to set this up the same way. We're going to take these two, we're going to split them up at the top, capital T, lowercase t. We're going to take these two, capital T, lowercase t, on the side. Drag and drop again like we did before. Capital T gets pulled down. Lowercase t gets pulled down. Then we're going to do the other way. We're going to go sideways. We're going to take this capital T, it goes here. And I always put the capitals first. So when you see a situation where you're pulling it down and you got a lowercase, you always want to make sure that capital goes first. It's just a good habit to have. So we had the capital T, lowercase t, and then we pulled it across. We're going to pull this lowercase t across. And what did we get? Well, our percentage looks a little different now if you've figured that out. We have 1, 2, 3 of these squares, which would be 75%, are tall. 75% of the offspring are tall. 1 out of 4, which is 25%, are short. So it's a 3-to-1 ratio. And that happens because this dominant trait, whenever you see that capital T, remember it's the dominant trait, the tall trait, it's going to overshadow everything in these squares, so it's going to be tall. But in this one, there's no capital letter, so the recessive trait is coming out, which is short in this case. Now, you can split it up again if you wanted to split it up by genotype. If you wanted to split it up by genotype, you have 25% capital T, 50% capital T, lowercase t, and 25% lowercase t. That's if you're looking at genotype, okay? Genotype, which is looking at the pairing of the genes, which is the letters. Phenotype, which is down here, which is looking at what trait it is, what's the physical trait, tall and short. Okay, so that's my example for crossing two heterozygous traits. All right, for this example, we are going to cross a homozygous green pea plant with a homozygous yellow pea plant. And in this case, we're indicating that green is the dominant trait. So just looking at that, we know that homozygous means that it's the same two letters. So homozygous green, and green being the dominant trait, we are going to represent that with G's. We'll do G's for this one. Capital G for this pea plant here. And homozygous, again, same two letters. Homozygous yellow, in this case, is going to be lowercase g, because it's the recessive trait. So we're going to cross these two. Make our Punnett Square. And let's get our two parents set up. So again, we are looking at a homozygous green, which we've already determined would be a capital T, green. And our homozygous yellow pea plant, which we've determined is yellow. And it's lowercase letters because it represents the recessive trait. So we're going to put our lowercase letters here, capital letters here. Same thing we always do. Pull down and pull across. Pull down. Pull down. Pull across. Pull across. Now in this case, it ends up being pretty easy because all of the offspring end up having the same genotype, which means they have the same phenotype. So all of the offspring, 100%, are green, and 100% are capital T, lowercase t. So both the phenotype and the genotype are 100% in this case because all of the boxes ended up having the same two alleles in them. So that's when you get a purebred dominant and a purebred recessive, this is what you end up getting. So it's 100% and 100%.